Our speaker is Professor Mohanty, who is professor at Stem Cell Facility Center of Excellence for Stem Cell Research at All India Institute of Medical Sciences in New Delhi. Her area of interest is stem cell biology, immunomodulation, exosome as cell-free therapy, and tissue engineering. She's a PhD from AIMS in 1996 and then postdoc fellowship from Mount Sinai, New York, USA. Her ICMR International Fellowship is from Kyoto University, Japan, and she's a member of the Institutional Committee for Stem Cell Research for different institutes, including National Institute of Immunology, and she is a reviewer in many journals, including international and national journals, as well as in the editorial board of Journal of Review. She published 120 research papers with an average of 8 papers per year, received 1 patent, and she filed 7 patents already. She is author of 6 book chapters. And today she is going to talk about Unani-based indigenous approaches to apprehend and bridge cancer progression. Over to you, Dr. Mohanty.
Thank you so much. I'm really glad to be here in Duke campus again. I have done my graduation from Duke, and it's like a homecoming here. Thank you so much, organizer, and especially Dr. Alok, for giving me this opportunity to be in the present of this August gathering. So at the onset, let me make one disclosure. Fine. I heard my previous speaker, Professor Satyanarayan, and I think there are more speakers who are going to come, and they are expert and luminaire in this field. And I am a totally novice. I don't know how come I am here. I think now I am feeling myself a misfit. But my area of expertise is stem cells, so I would like to blend my area and a little bit touch upon why I was interested to move into Ayurveda and our conventional drug, and what is my aim. So I hope you will bear with me. And this is the institute from where I come. And in my talk, I'll be a little bit talking about the stem cells, what are the translational efforts we have made using stem cell research and stem cell technology, and of course how we can make stem cell really more potent as an immunomodulator. Like latest COVID pandemic has shown, it's something to do with our body immunomodulation, and we were thinking that how we can modulate stem cells for its activity. There is another component of cell which is called exosomes, that means cells secrete out small bodies, and we were very much interested, can we leave aside the stem cells and use these exosomes for human use and diseases. And last one, I will be talking about the organoid as a model for cancer and how we are going to utilize that. This is the latest research my lab has initiated. Let's go back into the history. So I have made my presentation keeping the budding scientists and the young faculty who are here to motivate them, and also to show them that how we at AIMS initiated this field of research. So when you see the history, so these are the 3 gentlemen who got the Nobel Prize. And in 1919, we all know Sir Donald Thomas got the Nobel Prize when he worked on mice model and gave irradiation into the marrow and tried to see that if you do bone marrow transfusion into the irradiated mice, the mice survives. So that means something there in the bone marrow which is making these animals survive, which was the stem cell present in the bone marrow. Then Professor Yamanama, he got Nobel Prize for making your adult cells from the skin behave like embryonic stem cells. Per se, embryonic stem cells, you can only take it from the embryos. But what he did, he took biopsy from a human body, and these are the skin cells. He introduced 4 important factors so that these fibroblasts are turned into embryonic-like stem cells. The future of these iPSCs, which are called induced pluripotent stem cells, is either you can grow them in the lab, that means they will not die. If you grow your skin cells in the lab, after a few passages they die. But these skin cells which have been transformed by 4 factors, now it has been reduced to 3. They can grow for long period of time. You can do gene correction in them, and then you can make different type of cells using differentiation protocol, and then you can give back to the patient. So why I am talking about gene correction, there are certain genetic mutation. Right now in India, we are using this technology to correct the factor 8 and 9, which is associated with hemophilia. So we are correcting factor 8 and 9 in these stem cells and then giving it back to the hemophilic patient. So this is one of the examples. So in AIIMS also we started our work, and I'll just give you in the highlight that whatever work which we did in the lab, how it has gone into the practices. So this is the overall scenario worldwide where people are using stem cells for the therapy. If you see here, the green area is showing the type of diseases which are recruiting patient using stem cells. These are mesenchymal stem cells. I'll tell you later about them. Red means they are completed. Yellow means they are not active in recruitment, and some of them have been terminated. But this is the latest from 2015 till now. The different type of diseases you can see here, from COVID-19 to all the area including cancers and others. In every spectrum of the disease and the disorder, these stem cells have been investigated. So this is our place at AIIMS where we do stem cell research, both basic and clinical. We started our work in 2005 because there was some paper in 2003 which said that not only in cancer, there are other certain degenerative diseases where stem cells can be used. They were talking about two papers in Nature and Cell about heart disease. So we initiated our work, and then we moved to the phase 1 of Center of Excellence. We did multicentric trials. Then we had our basic research, increased our clinical collaborations, basic collaboration within India, outside India. Then we moved towards stem cell based product in terms of commercialization, and now having public-private partnership. So this is the overall scenario of clinical research. I will not focus on that. I'll just give you a snapshot of 2 clinical research. Just to mention it here, there are some guidelines which we have to follow, which are pertinent to India. So we follow those guidelines in our place. And all the work which we do in terms of stem cell manipulation and expansion is done in a specialized lab. These are called good manufacturing practices. So these are the few snapshots of our GMP lab. The first one is the repopulation of the eye, that means the ocular surface destruction. So if you see at the eye, you can see that it is very clear, and you can see the light is able to pass, but if there is some injury in the eye, your cornea, which is the transparent layer of the eye, will get occluded. It is very turbid. So eyes cannot perceive the light, and the patient is virtually blind. So we try to use a special type of cells which are called limbal epithelial stem cells. We try to isolate them, grow them in the lab, and with the aim, can we again bring back those clarity in the cornea. So I'll just show you here, this is the limbus area which I was talking about. This limbus area, the nature has given you stem cells. So if you see the another snapshot of the limbus area, this is the limbus, this is the conjunctiva which I call the brown area, and this is the clear cornea. So limbus area, whenever you blink your eye, few cells are lost. From the limbus area, stem cells migrate and they move over to the cornea. So we took the advantage of these cells. We took a biopsy from the limbus area, bring it back to the lab. We grew these limbal epithelial stem cells, made a sheet and transplanted back into the patient. So again, I'll show you, one of my students, Shweta, she did the basic work. We initially started with cadaveric donor. We got the limbal ring. We isolated from the ring a small piece of biopsy which contains limbal epithelial stem cells. So this is a small piece. When you grow them in culture, you can see here the whole sheet is growing, and then we characterize them using whole panel of immunofluorescence and specific proteins. So I'll not go into the detail. We do the corneal specific markers and also the stem cell marker, which is ABCG2. When we are sure that they are maintaining their phenotype and genotype, then we take the permission from the ethics committee to do the transplantation. So you can see here, this is the normal eye. If one eye is affected, the other eye we use as a biopsy. 2 dots are made and we take around 2 millimeter of the biopsy sample from the donor eye. This work was done along with Dr. Adhika Tandon, who is a surgeon in ophthalmology. The small piece you can see here, it is cut and sent back to the lab. Then in the lab we grow them over amniotic membrane and we give them a 37 degrees centigrade, and within 14 days they make a whole sheet, and this sheet is given back to the surgeon. So this is the recipient eye. You can see the cornea is totally opaque here, and they first clear the surface and then they put this sheet which is containing limbal epithelial stem cells and they suture it. So after some time you can see that the vision is exactly as the previous. This is the donor eye. I want to show you this picture that taking a biopsy from the donor eye does not do any harm to that eye. This is the recipient eye, the lower panel, and after 4 years you can see that the vision is exactly 6 by 6. This is a 17-year-old female with a chemical injury. This is another one which is a 16-year-old male, and you can see here the upper panel shows the donor eye, lower one is the recipient eye. At 17 months the cornea is clear. So we have done more than 1,000 cases now, and we are going to soon going to offer it as a routine therapy because it was done under a project mode. I'll just skip this one. So second one is vitiligo. So this is again a very specific problem to India because we are dark-skinned, a majority of us, and loss of pigmentation in the vitiligo causes some inhibition, especially in the younger population. So this is a case of a vitiligo. As you can see, this is loss of melanocytes, which is the pigmented cells. So the routine treatment in our aims is like you create blisters or you make skin to come up under pressure, and these are suction cups which are placed over the thigh, and after some time, maybe after one hour, you have to cut these topmost skin layer, and this topmost skin layer is transplanted back into the vitiligo site so that normal melanocytes can repopulate that area and you get the skin coloration. So it is very painful. So that means you have to sit for some time, and when you cut these skin topmost layer, there is a scarring problem. So one of my students, Anil, he read one paper and he came to me that hair follicle contains some of the stem cells. So this is called the bulge area of the skin, the central one, and they contain stem cells, and they also contain melanocytes. So that's why your hair is of particular color because of these stem cells. So then we discussed with our dermatology counterpart. We got some hair follicle cells. You can see here, these are the hair follicle cells, and under the microscope these bulge area can give rise to small cells. These are bulge stem cells, and these cells can be differentiated into melanocytes. So lower panel is showing the differentiation of stem cells into melanocytes. So after again characterization, we went ahead with transplantation. You can see here, this was done in collaboration with our colleague, Dr. Sumesh Gupta. This is one patient, and after transplantation of these hair follicle stem cells, you can see the repigmentation. So we did it in 15 cases, and we did follow-up of approximately 4 years, and they were pretty stable. So we need to move forward in this area again, because it was showing some very promising results. So as I said that my lab is focusing on mesenchymal stem cells. So what are they? So if you see the bone marrow, majority of them are hematopoietic stem cells. So these cells are very useful for cancers or treatment of blood cancers. But there is another type of cells which is called mesenchymal stem cells. It's here. So they are the feeder cells. They give support to the hematopoietic stem cells. They are much less as compared to hematopoietic stem cells, but they are very beneficial. In the sense, every major tissue in our body has mesenchymal stem cells. So my lab, they work on placenta or the Wharton jelly derived stem cells, the bone marrow stem cells, and adipose stem cells. So the Wharton jelly, adipose, and bone marrow. So these are the 3 types of mesenchymal stem cells my lab is working on. Another advantage of these cells is, again, they can differentiate into multiple type of cells. I don't know, this picture is not coming, okay. But they are a HLA class 2 negative. That means you can easily give mesenchymal stem cell from one person to another, or you can pull together 5 individuals mesenchymal stem cells and can give without any matching. So this is one of the greatest advantage of mesenchymal stem cells, whereas hematopoietic stem cells need vigorous matching before transplantation. So this is the mode of action of mesenchymal stem cells. I will just give you a few examples in some of my slides. So these are the 4 areas which I am going to cover in a few slides in terms of how do stem cells differentiate, what is immunomodulation, which I talked about, which is now the recent buzzword to combat major diseases. So this is the study, like what is a comparative study which we did, like how mesenchymal stem cells can do the immunomodulatory effect. Here we again took mesenchymal stem cell from adipose tissue, Wharton jelly and bone marrow, and we saw that all 3 type of mesenchymal stem cells and their product, which is secreted in the conditioned media, can have potent immunomodulatory effect, and these are via IDO, HLA-G and PG2, and these are the molecules. I'll not go into the details, everybody knows the native immunobiology. They are the potent molecules by which major immunomodulatory effect occurs. So if we compare 3 sources, the bone marrow, adipose and Wharton jelly, we found the Wharton jelly, which is derived from the placenta, has the maximum immunomodulatory properties. So then we also check their functionality. So we check in the lab. So how do you check the functionality? That means you co-culture the MSC along with your patient's allogenic form peripheral blood mononuclear cells. So when we did the MLR, which is called mixed lymphocyte reaction, there also we found that it was a potent immunomodulator and it was able to decrease the proliferation of these cells, that is by the Wharton jelly. All of 3 of them showed potent immunomodulation, but again Wharton jelly showed the maximum immunomodulation. Then we thought that can we modulate the parent cell, that is the MSC, can we give some signal to these MSCs so that they increase their immunomodulation? So this was our hypothesis, and there we thought of some molecules. This paper is again published. So the molecules which we chose was dexamethasone, progesterone and interferon gamma. So because these are the molecules which are upregulated during some disease scenario, or maybe chronic or acute, so we chose these 3 molecules. So in a nutshell, I would like to say that this was the overall plan. We exposed MSCs to 3 drugs, that means dexa, IFN and progesterone, and we took 3 types of MSCs. We calculated the dose of these molecules and how much time do they need to do the immunomodulation, and what are the effect on again HLA-G, IDO, PG2 and HLA class 2. We did multiple parametric analysis, that is gene level, protein level, and also the functional assessment in terms of mixed lymphocyte reaction. So in a nutshell, I will just say that again there was very marked immunomodulation effect by priming these MSCs with 3 molecules, and we established different doses, and now we are going to carry forward this work in sclerosis and other immune related diseases. I'll just skip that. This is again the same with DEXA. Then there was one very important factor. Whenever we grow these MSCs, we grow under normoxia. But when you see inside the body, it is not normoxia. So I don't know why that slide is not coming. Inside our body, there is a gradation of oxygen. The bone marrow oxygen concentration is not 21%, which is the atmospheric oxygen concentration. It is around 1 to 5%. So we tried to grow these MSCs under hypoxia, and we again did this panel of tests, and we found that giving stem cells the hypoxia again increased their immunomodulation. So these are some of the papers on them. So this was our latest finding, and we are going to again do in-depth analysis that what is the mechanism by which hypoxia is increasing the immunomodulation of stem cells. Another area of research is mitochondria. So one of the important aspects of mitochondria is that it is a powerhouse. So we call it powerhouse because it produces ATP. So another important factor is that we all inherit mitochondria from our mother. So whatever mitochondria we have it inside us is from maternal transmission. So we said, okay, let's see what happens in terms of mitochondrial health of stem cells. We did a small experiment. We took epithelial cells from the skin. We destroyed its mitochondria. Then what we did, we brought together a stem cell in the close proximity of the skin cells. And we found that this MSC is making a tunnel, and through this tunnel, the mitochondria which are healthy, present in stem cells, are being transferred to these dying cells. Because if you kill a mitochondria, cell is going to die. So this is, I am showing you in a real time. This is the skin fibroblast. All mitochondria is damaged by rotenone and it is stained green. Then we have MSCs, its mitochondria is stained red. And what happens when you co-culture, you can see the green is turning into orange because some of the mitochondria is being transferred by these nanotubules. I think the picture is not very clear, but you can appreciate that this green fibroblast is turning into orange. And this is a video you can see, if it runs, sorry. This was a nanotube, it is not working, and this was a small mitochondria which was traveling from the donor cells to the recipient cells. So that means stem cell can also be a good potent of mitochondria donor, and again there are few papers who are working on isolating mitochondria from stem cells and giving it in different diseases. Another aspect, the third and the last one, is the exosome I talked about. So MSCs, these are stem cells. They secrete some of the growth factor and cytokines which comes into the bloodstream, and then they are distributed throughout the body. But the stem cell also secretes some of the vesicles which are pinching from their cell membrane, and these exosomes, they contain some of the mitochondria, microRNAs, mRNAs, cytokines and growth factors. So what is the difference between them? That they are much more sturdier. They are very, very sturdy. They are not affected by the temperature and any other adverse effect. So in our lab, we tried to see that what is the effect of these exosomes which are derived from the stem cells on liver injury. I don't know, the one slide is missing, so which was the overview of that. I'll just skip that. So this is the picture, like when what we did, we created a liver fibrosis model by giving a very harmful drug, and after that we injected these exosomes, and we found that at day 7 there was reversal of liver fibrosis in these animals, so in these mice, which were confirmed by HNE, mesenteric and Cyrus red. These are the marker of collagen deposition in the liver. I think there is some problem in the slides. Sorry for that, some of the slides are missing. The ultimate message was there was significant decrease in the inflammation in the liver. There was decrease in the fibrosis, which was the cause of liver fibrosis, and there was regeneration of the liver. We could find some hepatocytes in the liver. This is another study I would like to highlight. Here we have tried to see if these exosomes can reverse neuroinflammation. So we have taken the example of Japanese encephalitis virus. We created that animal model and we injected exosomes into them. We found if you take JV infected rat, it dies within 3 days. But if you injected exosomes, these rats and animals, they survive. Can I have 2 minutes more? Or time is up? Okay. So this is some of the features I want to see here. That means these are the neurosphere. That means if you give JV, these neurosphere are small in size. But if you give Exosomes, the size is maintained. That means neuroinflammation is much reduced in them. This is again the animal studies, and this shows the survival curve of the animal. This is the mock, that means all the animals they die, but if you take this, the green line, these are infected animals, and if you give EV, they survive. Again, by immunohistochemistry, we found that the neurons present in the brain are also very much healthy. These are all slides showing the regeneration in the brain also. Again, we did the immune profile of the animals also. We found that the inflammatory immune system also showed that there was enhancement of CD4 and neutrophils, and there was decrease in neuroinflammation. That means these EV can cross the blood-brain barrier and can reach into the brain. So that was the biggest finding which could find, because stem cells, they do not cross the blood-brain barrier in the brain. So this is one of the picture. The last animal is JV infected, but given exosomes, it is very healthy and it is just jumping out of the tray. And this animal is JV infected and it is hardly able to move. So this was the stark difference we found in these animal studies. I'll just skip these slides because of the time. I'll just go what is the way forward. This is the cancer treatment which we are going to start using the organoid model at AIIMS. So why organoid? When you use 2D system, it is different from the 3D system. We are trying to make a 3D system which can mimic the cancer. So here are some of the example of, this is a brain organoid, and this is again a brain organoid which has got the retinal cup. So retina, they are black in color, so you can see the brain organoid with the retinal cup in a dish, and these are intestinal organoids in the culture. So likewise, we are trying to make these organoid using breast cancer as a model. You can see here, these are some of in a suspension which we have made. If we take 500, the size is small. If we increase the number of cancer cells, the organoid is larger in size. These are again pictures, some of the pictures are missing here. This is again a breast cancer organoid, and we characterize them, and now we are using different drugs to test it. Sorry about some of the pictures are not coming because it's not compatible. I made it in the Mac. So because of some publication which has come out in Ayurveda and Unani, so I was very much interested that why not I combine my knowledge of cancer stem cells using organoid model and try to test these compounds. This is just the initial studies, and hopefully we can take it forward and come out in terms of scientific validation of these, so that we can tell not only to Indian people but also universal and global person about the effectiveness of these Ayurvedic and Unani drugs. So these are some of the drugs which we have tested, and I think I should not dwell more upon them. We have shortlisted these 4 drugs. My slides are not coming. It's coming? Yeah. No. It's delay? I don't know. So we tried these 4 drugs at different combination, and we found that, I don't know, yeah. It's cross. It's not taking. I think there's some compatible problem. I apologize for that, yes. So I made it in Mac, it's not coming. I'll just tell you, like, we made these organoids, which is in a 3D structure, and we compared different drug doses on them, and there was a great reduction in the size of the organoid. And we also checked their toxicity. So they were not toxic to them as compared to the conventional paclitaxel and other drugs which is used for the routine breast cancer. I'm extremely sorry for that. So the take-home message is that it's an evolving field. We need to do a lot of research to validate our data and then prove to other people who always see our Ayurveda and Unani in a very skeptical manner. So I think this should be the end of my slide. I would like to thank all sorts of funding and my collaborator. Extremely sorry. And thank you slide is also not coming. I thank you so much. Thank you so much. I don't know why thank you slide is also not coming. This is my team. If you can see, and if I can bring my thank you slide. This is my team. And thank you slide. Thank you so much.
Thank you so much for the fabulous talk. It was very informative, and I'm from the stem cell field, so it was nice to know many new things from you.
Thank you so much.
The talk is open for discussion. If there are questions, one or two questions we can take quickly. Uh,
Thank you, ma'am, for a nice talk. Ma'am, I have one doubt that because we have talked about how mesenchymal stem cell help in regeneration of damaged tissue and in immunomodulation. But if you want to target the cancer stem cell, is it that what is the main difference between the cancer stem cells and the mesenchymal stem cells? Are the mesenchymal stem cells are the part of cancer stem cells or?
I think it's a very important question. It's also a very difficult question to answer. It's like cancer stem cells is again a very hypothesis. People are still debating, is it the cancer cells which acquire the property of stem cell, or is it some of the stem cells which are gaining the properties of cancer and becoming cancer stem cells. But we do know in a cancer, some of the major cancer cells are being killed by radiotherapy and chemotherapy, but some cells are very refractive to any therapy, and those cells are termed as cancer stem cells. So what we are trying to do in our lab are using cancer cell as a model to study cancer stem cells. And you all know that mesenchymal stem cells are also part of making cancer cells malignant cells, because they are very migratory cells. So our hypothesis is cancer stem cells are modulating the mesenchymal stem cells in the tissue, making them behave like a cancer cells, and then they get detached from the site, go into the periphery and cause secondary metastasis or like they spread the disease. Right? I hope I answered.
Okay. Then one question again, it is from your exosome parts that the exosome released from the mesenchymal stem cells and they help in the liver regeneration. So what factors actually the exosomes carries inside, so they help in
Again a very nice question. So as I said, these exosomes are parent cell specific. So whatever factors are there in the parent, these exosomes package them inside, which include microRNA, protein, some DNA fragments. So we are working on microRNAs. So we have identified few microRNA, I didn't show you that, which are liver hepatocyte protective, which causes hepatocyte regeneration and also causes the fibrosis to reverse. So there are few microRNAs which we have identified, which were there in MSC exosomes.
Thank you.
We'll have just two questions only. If you have more questions, you can get in touch with Dr. Mohanty later.
I request